Hello, my name is Helen Chambers and today I'm going to be talking about my current research focused on addressing the question, why big brains? Specifically looking at carnivore brain size evolution. To get started, I wanted to highlight a few key themes in brain evolution literature. The social brain hypothesis, suggested by Dunbar in 1998, is perhaps the most influential hypothesis in the field. It suggests the cognitive demands of tracking, maintaining, and repairing social relationships to be the main driving force behind variation in brain size. Originally, this was suggested in primates. However, the theory has also gained support in other species. In carnivores, considerable evidence comes from research on spotted hyenas, whose societies appear to draw parallels with Cercopithecine primates in terms of complex social societies and certain life history traits. Group size became the most commonly used proxy for sociality. However, greater attention is now often paid to the presence of pair bonds, looking more at relationship quality, not quantity. Ecological hypotheses mainly involve investigating the cognitive demands associated with foraging. This is because foraging is considered demanding due to the pressure of managing, processing, and remembering spatial and temporal information about resource availability. This has resulted in many studies with a particular interest in diet, bone range, foraging techniques, and behavioural responses in a changing environment. Despite the current interest, the role of diet has only recently become of interest again. This is because when Dunbar proposed the social brain hypothesis, he discredited the role of ecology, finding no support for the ecological proxies investigated. So where are we now? More recent studies have failed to find a statistical link between brain size and measures of sociality. Instead, strong phylogenetically corrected comparative data now suggest diet better predicts brain size in both primates and carnivores. So why is there this disparity in results? These contrasting results are thought to be the consequence of increased sample sizes, updated data, and improved statistical analysis. However, another problem which has hindered comparative analyses of brain size is deciding on the correct brain measure. Certain examples include using residuals as relative brain size or calculating encephalization quotients. However, there is little clarity in the literature regarding the correct measure. So to address the current disparity within the field, this is what we did. We collected data on both whole brain and regional brain volumes, specifically the neocortex and cerebellum. We collected data on social, ecological, and life history variables. We chose to include the life history traits as they are thought to exert influence on cognitive evolution and the growth of large brains. We ran a series of different models which range in complexity, ranging from an exclusively social model to a more complex combined model. We also used different brain measures in order to test how uniform the results were across different methods. Critically, we used PGLS, which allowed us to run regression analyses while accounting for phylogeny or relatedness. The table shown here includes a sample of our EQ scores, where a score below 1 equals a brain size smaller than expected for body size, and a score above 1 equals a brain size larger than expected. So what did we find? Whilst past work found support for the social brain hypothesis in carnivores, in our study the relationship is actually negatively correlated. This result is similar to more current research which found no support for the social brain hypotheses in carnivore. We think the contrasting results could be due to the fact that sociality in carnivores appears to be more limited to a select few taxa, such as spotted hyenas and lions. Thus, while sociality evidently plays a more important role in primates, for example, this is less common in carnivore species and therefore does not hold the same importance. Interestingly, the influence of sociality was restricted to the cerebellum models, which is consistent with current research by Rob Barton suggesting the cerebellum is the brain region involved in social intelligence. In contrast to previous research, we find evidence for a negative correlation between dietary breadth and brain size. This contradicts the current idea that dietary generalists possess larger brains and superior cognitive skill sets than those with more specialized or limited food variety. The dietary breadth scale ranged from 1 to 6 based on the number of different food types within the diet. Therefore, we propose this result is a consequence of those who are classified as meat eaters, whose dietary breadth is limited to 1 or 2 categories, thereby producing this negative correlation. Consistent with several life history hypotheses, There are several negative correlations found with brain size in carnivores. This reinforces support, especially for the expensive brain hypothesis, which suggests in order to develop a large brain, there must be a trade-off with another expensive organ or function, which here seems to be reproduction. When paired with an increase in maternal investment, as evidenced in the literature, this bypasses the developmental constraints of producing a large brain through reduced fertility and slow maturation times. Critically, we found using different brain measures produced contrasting results. This highlights how important the method of choice is. In addition, the inclusion of specific brain regions uncovered further associations that were not present in whole brain models. For example, the social associations were only present in the cerebellum models. Therefore, without including this brain region, the influence of this association would have been missed. To conclude, both ecological and social variables appear to be influencing brain size in carnivores. This requires greater investigation. Life history traits reveal evidence for the transition to slow life histories, which work towards facilitating the production of big brains and bypassing the cost of expensive brain tissue. Future studies should give careful consideration of brain size measures, as different methods are found to produce different results. Future studies should strive to integrate multiple variables, fully encompassing all the potential variables influencing brain size, as we have here. This includes careful consideration of body size correction factors, which for carnivores, we recommend inputting body mass as a covariate. In addition, researchers should investigate both whole brain and specific brain regions, as inclusion of such may reveal further associations. I would just like to say thank you to my fantastic supervisor, Dr. Sean O'Hara, my wonderful co-author, Dr. Sandra Helstab, all the researchers who helped make the study possible, and thank you for watching.